Hi everyone, this is Tony from Junyan University. During this Biology 110 presentation, I want to discuss our third lab, the cell division and basic microscopy. The reason I want to talk about this lab because it is very interesting and I really want to talk about the connection between the real world and this lab. Okay, this is the outline of what I'm going to talk about today. First, some background information of microscope and cell division. Second, I will review the process of this lab experiment. Third, I will analyze the real-world connection of these experiments, which I'm most excited about. In the end, this video covers a thank note to whom I want to express my thanks. Okay, let's go to the part 1. I will introduce microscope first. Microscope is a way for us to visualize the cells because cells are usually too small to be seen by our naked eyes. There are usually 4 types of microscope that are commonly used. First, light microscope. Second, electron microscope. Third, scanning electron microscopes. Fourth, the transmission electron microscopes. Since we are only going to observe basic cell division, a light microscope is enough for us to observe that clearly. So next is the introduction of mitosis. As for eukaryote, there are 2 types of cell division, meiosis and mitosis. What we are going to talk about is mitosis. It's a way to pass genetic information to next cell generation and keep genetic stability. The mitosis contains 4 phases: prophase, metaphase, anaphase, and telophase. However, there is a preparation before mitosis. The interphase, containing 3 subphases, G1, S, and G2. In prophase, chromosome appears, nucleus disappears. In metaphase, which is the best time to discover the cell circle, chromosomes line at the middle of the cell. In anaphase, 1 chromosome split into 2 chromosomes and thus the chromosome doubles. In telophase, the cell plate appears, thus divide the cell into 2 daughter cells. Okay, let's go to the part 2, the process of the experiment. In this experiment, I use the root of garlic to observe the cell circle. The garlic root tips are soaked in the fixed tip solution at first, and after minutes, I dry them by using gentle violet. And minutes later, the dried garlic root tips are pressed to form a monoid layer because in that way, without overlaps, we can observe them much more clearly. And in the end, I observed the different cell phases through light microscope. Please enjoy the photo I take. Okay, let's move on to part 3, the connection between the real world and this lab experiment. We can determine the time of different phase by analyzing the proportion of the cell number of different phases, right? So I found that the interphase takes up most of the time of the cell circle, approximately 7/8. So if we want to speed up or slow down cell circle, it is very important and efficient to take advantage of the long period of interphase. On the one hand, if we want to speed up the cell division and generate more cells at a time, for example, there is a patient who suffers from large area of burn, therefore he badly needs the skin to grow faster, right? If we can develop some enzyme or catalyst that can speed up the interphase, then it's gonna be a breakthrough for the patient to survive, right? On the other hand, if we want to make the cell circle slower, for example, we want to inhibit the cancer cell to grow, right? So we can probably develop a toxic that targeting at cancer cell interface, right? So this is what I think very important with potential in the future biology research. Last but definitely not least, I want to leave a thanks note. First, to my parents, I really appreciate that your full support to me to go to DKU. I know that means to you I have to go abroad in the future, which brought tons of miss. But intern DQ do give me plenty of opportunity to discover and explore, and I'm so proud and happy that I met so many excellent peers and professors. And I also want to say thanks to Professor Li, Professor Mian, and Professor Su Hua, because you really give me a new side to see biology, which is totally different from that in high school. And during my labs, I have a lot of questions, right? But you are so patient and caring to answer my questions. So many thanks. And by the way, you guys are so kind. I like you guys so much. And I also want to say thanks to my Think Pair Share group. I made a lot of discussion and achieved extra credit, right? When I discuss questions with you guys, I believe you can feel it too. We're making progress together. Hope to meet you guys